It's interesting to me that since showing an interest in science, specifically biomedical sciences, and starting a degree in biomedical sciences, I've kind of been of the mind that the more I learn, the less I should talk about what I learn. And it kind of goes hand in hand with things like, my odd medical issues becoming a lot more real, like over time as things went by. Like there was a point in time when certain conditions I could have I don't know, maybe I was mad and it was figments of my imagination. And then the more I spoke to professors and such, it got realer and realer over time. And that caused a bit of a weird cognitive shift in my mind that they are learning things from me as much as I'm learning from them. And the more I talk about these things online, I may be spreading misinformation because some things are cutting edge, particularly with the post-COVID issue, because one of the GPs that, I've been discussing that with actually helps to run long COVID clinics in this part of the country and trials for dealing with, certain long COVID issues. And, I've got my theories on things, and I've made the choice to kind of keep most of that to myself now, because even though I'm very limited in what I can do for myself, the actions of seeking out specialists and talking to them and having to explain the issue provides a really uncomfortable amount of anxiety. It makes it realer and realer, whereas I'm more comfortable dealing with a certain amount of suffering at home. And, with all of those issues, I've had some success, in finding solutions. And this post-COVID one is still proving to be the most difficult, but there are glimmers of very specific combinations of ingested things that can modulate the symptom profile. Anyway, the thing I want to say is that it's funny, such as when doing the recent video on visualizing proteins in Blender. In that video, I make a rather, excuse me for saying, modest point that, I don't know much about this stuff. And, ooh, wow, look, isn't it so interesting, this website with all this protein databanks? And it's been really funny because after that, I visit the Blender Science Discord server, and I'm sure some of them will watch this and listen to this. And I see a lovely post from someone saying, I shared this feedback in Curtis's video about different ways to see proteins. Curtis doesn't know anything about proteins, and he was just doing it to cope with his long COVID issue. Forgetting the fact that, I've done courses on comparing the genetic structure of influenza and SARS-CoV-2. And I did a really hefty course on antibiotic resistance, and I found that really interesting, especially beta-lactamases, because it's really interesting how, that's the granular level of mutations for getting around antibiotics. It's weird looking at something so specific in that way and going, I can visually understand evolution now. Beta-lactamases are enzymes, a form of protein, that modify or basically destroy a beta-lactam antibiotic like cephalosporin because it breaks this molecular ring. And bacteria both did figure out and didn't really figure out how to do that. Evolution is a really weird thing. You can think of it very emergently. There's a fantastic video by a Harvard medical school experiment where they divided a huge petri dish, equivalent type thing, into different sections with increasing gradients of an antibiotic present. They had E. coli, and they started it at the edges, and then they watched it grow, and then when it reached an antibiotic ban, after a little bit of time, not even that long, the new mutation would branch out. Mutations can convey advantages, but also disadvantages, because oftentimes there's a trade-off. And we can think about that in other areas. There's a great theory on why do we have such fantastic language abilities, but we don't have the short-term memory that's demonstrated by other members of the primate family who are fantastic at short-term memory, particularly visual memory. The idea being that we've had a trade-off in the development of the brain, focusing on one area while losing out on another. The same thing tends to happen on all scales of evolution. Not uniformly, but it's one thing to keep in mind that, when thinking about epidemics and stuff, people are often scared at the idea of mutations, as in, this thing is going to get progressively worse and worse and worse, and we're all going to die. But life is not really that simple. Things tend to homogenize in some way. So oftentimes where an advantage has been conferred, a weakness may also open up at the same time. Just one thing to keep in mind. But we're not going to go into that because I've got the British mind where the more you talk about expert things, or the more fancy terms you use, I think the more of a cunt you sound. Which is why, the more I'm able to talk about things, the less I'll do it, which has the unfortunate side effect of, because you don't, project knowledge online, people think you're an idiot. So it's weirdly paradoxical, but I'm okay with that. Anyway, I wanted to show you through the little lab corner now, because there's been some changes. Now, everything you're going to see here in the lab corner is stuff that I've in some way shown like in the past, but none of this is professionally taught. Okay, I'm doing the degree at the moment. Whether or not I continue with the full course of the degree depends on like how my work goes in the future, and because I'll take it year by year and reassess how things go in terms of balance. But one of the impetuses for doing like home, very simple hematology in the form of blood smears comes from a few different reasons. You might think, is that like a weird obsession, like, you know, blood smears, etc.? Because, you know, I've done previous videos that people have found a bit odd to watch in the past. And yeah, there are a few reasons. When I was like, you know, a kid, well, I've got like 3 blood conditions. So I was investigated a fair bit in London, and no one believed me when I tried to explain how like unbelievably painful like the blood tests were for me, or no one really cared. So it was very much like, oh, I hated being in school, and then you'd get dragged out of the place you hate the most into the place you hate the most a little bit more to get tortured effectively. And then what do you do after that? Oh, you get like dragged back to school again. It was not fun. There was a funny moment where I kind of snapped at a doctor as a kid, but that's like, you know, a story for another time. So I always wanted to get over this fear of like blood pricks and blood tests and stuff like that. It took me ages to get over the fear of blood pricking, but I've made like huge progress now simply from this endeavor. But also during the corneal neuralgia issue, I had a sense of desperation that I've never had before for a medical problem. And, you know, a lot of people have committed suicide from corneal neuralgia, usually after laser eye surgery, where ophthalmologists do not communicate the risks, and adequate research has not been done regarding people, not everyone, but a fair number of people having inadequate nerve regeneration capability for their cornea after it's been lasered into. In particular, and this is my theory now, so this is not proper information, but it seems as though changes in the immune response, or an unpredictable immune response, seems to have prevented or suppressed like nerve regeneration abilities. Now, I really shouldn't speculate like this, but steroids were what got me out of it that time. They don't work for everyone, but one of the possible treatments for like nerve regeneration is autologous serum tears, which requires producing these tears from your blood plasma, basically these eye drops from your own blood, because your blood is dense in these growth factors, and it's a perfectly valid and perfectly viable like therapy. The thing is, it was hard finding someone that would take me seriously, because when I was trying to explain the pain and the weird like nerve sensations I was having, most of the ophthalmologists had never heard of those sensations ever, like in their whole professional life. They'd not been trained on it. They didn't believe me. And the thing that really pissed me off more than anything was no one offered to refer me to someone else. Everyone was so proud, like, oh, they knew that it must not be anything because they had never heard of it before. I'm happy to say that finally I did bump into a now professor, then researcher. He's a professor of corneal regeneration now, which is a fitting title, who was the complete opposite, who told me that everything I was describing he had seen before, he had bumped into before, and that goes to show how powerful a research background is. So now whenever it comes to those situations, I always try to seek out the researchers, as in the people with actual intellect, not the people that just stumbled into a specialty because what they wanted to make money or they didn't have any other options going. You know, find the people who are truly curious and truly work on acquiring knowledge and using that knowledge to build new solutions. Anyway, being able to have a look at my blood cells, and specifically the white cells, would be a benefit to me. There are 2 conditions I have that can be monitored this way through microscopy. Like I said, I haven't been professionally trained on this, so what I'm doing is I'm effectively using substitutes for a lot of things. For example, I'm going to be using isopropyl alcohol as a fixative. And now there may be like specialists that watch this and go, oh, you shouldn't use isopropyl alcohol for fixative. I'm going to be using just methylene blue for a stain. People will go, oh, you won't get the full spectrum of leukocytes that way. It won't stain eosinophils properly. Like, a lot of these things are known. And like I said, you know, earlier about how if you don't project this kind of arsehole-ish amount of knowledge in every video, people assume that you don't know things. It's always been the same thing with Blender as well. You don't do things a certain way, people assume you haven't been informed of that way. And you're like, actually, I just like doing it this way. But also, I personally enjoy the process of figuring things out. I don't want to be a scientist in the future. You know, a lot of people might see, oh, you're doing a biomed degree. Surely you want to go into healthcare or science. Nope. I'm just interested in it. It's a possible backup if everything else fails, but I'm not going to let everything else fail. So let's take a look at the lab corner. And specifically, I have a job at the moment. Not a real job in terms of anything paid, but a family member has a child, and me with my, you know, weird predictive foresight, when they came around for a visit one day, I gave them a microscope slide in a sealed plastic bag. And I said, if you ever like accidentally, I don't know, clip your child's fingers when cutting their nails, or if they have like an accident and, provided it's safe, if at all ever appropriate, just take a quick dab of, like a cut on this microscope slide and just put it in the bag. Forget about it and then, just hand it to me next time we meet. And funnily enough, it happened. But, children are like little accident machines, and it is what it is. A small cut occurred. She remembered, oh yeah, Curtis gave me that microscope slide. Because I had previously taken a look at her blood as well, the mother and other family members. Again, not a substitute for any professional care, but, they are nice enough to donate a sample for my self-learning and also out of curiosity. I mean, a lot of people, they want to see what it looks like. And we have found some really interesting things. For example, a cancer chemotherapy patient in the family donated a small sample, and I call it like a forensic sample. It's such a tiny amount from a capillary prick, basically, which, again, an expert will watch this going, oh, but you won't get the full information from capillary. And I'm like, yeah, I know. You can get a lot of information. In the cancer chemotherapy patient, they had a lot of nuclear debris laying around, which makes sense. But their actual proportion, the differential count for the leukocytes was actually pretty brilliant, like pretty normal. And to me, again, just, my random theory interpretation, it was like the body is actually functioning well, but the chemotherapy is just having a suppressive effect. And touch like all the wood in the room, apparently that person is actually doing quite well now, like with their recovery. I don't want to like jinx anything. I don't want to tempt the universe. But it was interesting being able to look at cells and go, this looks like the body is trying. Do you know what I mean? It's a weird thing to explain. Anyway, let's take a look. All right, so here I am in the lab space, the lab corner. What I'm doing right now is I'm just checking that things look okay under this microscope. This one, by the way, is what I call microscope beta. I've got two microscopes. One is smaller and older, which I call the validation microscope, microscope alpha. And this one's beta. It's what I use for like actual assessments, actual detail studies of samples, in this case, obviously, blood smears. So I'm just taking a quick look at the young child's blood here, and it's clumped up in a lot of areas. What I'm doing with my arm off the left of the screen is I've got a little, pen tablet screen, and it's connected to OBS, which is connecting to the camera on the microscope. And I'm doing what I'm calling a digital hyperzoom, which is where I'm not quite at the maximum objective lens on the microscope, but I can push it much further digitally. So that's what I do if I want to look closer without changing the objective. I just modify on OBS on the side screen. When I look into the microscope through the eye lenses, it's slightly out of focus compared to the main camera. Now that's an issue that I could resolve because there's a lot of issues I've still got to resolve with this setup. So that's why it looks so janky at the top there, actually, because my printer kind of broke. My 3D printer, one of the extruder heads, needs a bit of repairing at the moment, but it means I can't do like PVA negative space printing at the moment. So I basically just like hacked together this thing. And what you're seeing me do now is I'm focusing on blood cells which are more weakly distributed, so that'd be like along the edge of clumped up areas on the slide, where you can see them more individually. And I'm looking at the boundaries of the cells. What I want to see is them being like plump, basically, completely round, not misshapen. Because oftentimes if you're seeing things like extreme kidney or liver diseases, or any kind of like protein formation diseases, whatever, you'll see the boundaries of the cells. They will look like spiky or weird or just unusual. Blood cells, or otherwise known as erythrocytes, have like a very typical biconcave shape to them, which you're very used to seeing in, artworks and visualizations. So before I do any staining to look at leukocytes, that's just what I tend to do. You can see the validation microscope screen here on the left. So we're further around the left side of the lab. And what I'm doing is just showing you here, like, this is a test slide that I use to take a look. It's an old sample of mine, but I didn't really log any data on it. So I'm going to take the child sample away from microscope beta, and I'm going to bring it over so I can start doing some of the staining. What you'll also see here is my kind of Huion screen to the left of the laptop. And I've got, different things on there as well, including a timer to help me just track time when I do some preparation. Now, like I said earlier, I'm using isopropyl alcohol as a fixative, and what this will do is denature the proteins to prevent the cells from lysing, which means it will stop them from like absorbing so much water that they burst. So it's important to do this, for preservation. Well, it's not really preservation, especially not with isopropyl alcohol. It's just it's more dehydrating than anything, but it will mean that we won't have a problem when trying to stain it. Otherwise, the stain may cause the cells to lyse, and that's because the stain has been mixed with distilled water, and it will just like run off the slide. So yeah, I'm just applying some isopropyl, and I'll leave it on there for a few minutes because you don't need too long. And this this side monitor thing, you know, with the stylus, it's a new idea I'm trying. I didn't have this before, but I actually quite like it. It feels quite intuitive. So now that time's passed, I'm going to be draining off the excess isopropyl. And I don't typically want the drops to fall where the blood is, so I try and get it to run along the edge a bit. Now, ideally, you might just let it air dry. You know, if you're not using much, it'll air dry quite quickly, but you might think, don't you want to be careful? Like, won't the blood just like run off along with the alcohol? Not really. You know, this is the thing about, again, not being professionally trained, but also figuring things out as an amateur is you can actually get away with doing like a lot of like lazy stuff and still get the answers you're looking for. So now I've put the slide under the validation microscope just to check that it hasn't all lysed, but we're looking at the clumpy areas where things have really dried out and there's multiple layers of the erythrocytes stacked up. And there may be like some coagulation. Coagulation, by the way, maybe I should like just hold it here. You might wonder how like your blood likes to clot up and seal gaps and stuff. And it's really interesting. You've got a whole bunch of these factors in your blood and proteins that are kind of inactivated and ready to be activated or like cleaved. So one of them would be like fibrinogen, which gets cleaved into fibrin. And this is like a somewhat sticky, I would use that word, thread-like substance that basically picks up on cells, things like platelets and other cells that kind of help to form this boundary, this barrier. Now, again, I'm no expert in blood coagulation, but it basically just goes to show that your blood is basically ready to be reactive and to clot up, basically, which is, you know, useful. The body keeps it in this optimal condition, whereas this, you know, constant life-giving liquid, if you like. And as soon as it's exposed to the outside world and these harsher conditions, kind of outside of a homeostatic set point, then it's going to start activating these backup sequences. So I was quite happy, you know, like the cells hadn't burst in this, so we can now proceed to the staining part. And, you know, I'm being a bit loose and lazy with it. Typically, you want to let it air dry more, but we're just going to get straight into it. So this is methylene blue. You might know it. I think they use it for swimming pools and a lot of other things, but it's a really good stain for kind of highlighting leukocytes. Not every leukocyte. And if you don't know, a leukocyte is just a white blood cell. So, you know, your typical immune cells in the blood. So I have little accessible samples in small vials like this, and, you know, I like to kind of make sure the top is dry. It gets sort of wet at the top because it can't evaporate from these vials. So you end up with this like condensation inside of it, but I like to dry that out when I use it, just to keep it cleaner when I plug it back up again. So an important note to make here is that this dye has been diluted in my vial with distilled water, which is why it's important to use a fixative. So I'm going to grab some up inside of the pipette, just a basic disposable pipette. And if there's air gaps, I want to make sure that, you know, they're not in there. And then I'm going to apply it. And it's basically just a matter of obviously dropping it over the areas we want to look at. Now, weirdly, you know, staining the thickest areas is not necessarily what you want to do, even though that's what I did this time. Because when actually looking under the microscope, you want to find the thinnest smeared areas. Typically, if you had a lot of blood as a sample, then it would be very easy to get that. You know, you'd put a drop of blood on the slide and let it run down, and there's all different kinds of things for doing that. But what I'm doing is more of what I guess we'd call like a forensic way, where a prick has been made, effectively just like pushing the microscope slide onto the wound, and then we're just letting, you know, nature and randomization present it on the slide for you. And then from there, you're going to try and find the best points for it yourself. So now the staining's done, I'm basically putting it into what I call the stain drain, which is just, you know, a little metal plate where I will try and get it off, and sometimes tapping it down on the plate, you know, helps the drops to fall. Same kind of angle as the isopropyl, and the stain will actually evaporate from that plate very quickly as well. Not like immediately, but it really won't take that long. Like I'll come back in the morning and it'll be gone. A little bit of air drying. Okay, so now we can do the validation check again. So I'm going to put it under microscope alpha. Now, because it's still wet, it's going to be harder to see because the light's going to like, you know, kind of refract through it in a bit of a weird way. But I can immediately see like a lymphocyte has been highlighted, which is a darker blue circle. It might be a bit hard to see on the video. So lymphocytes are like your typical white blood cells, the ones that you probably know the most, like T cells and B cells. They get, you know, I guess most of the attention, but they just show up here as dark blue circles. What this tells me is that this is, you know, perfectly fine. As it's drying out, it's going to be a good one to look at. Although what I'm noticing is in this, I can only recognize lymphocytes, which, oh, this is really cool as well. You can see the boundary. between the dye and the undyed areas. So you can actually physically see how some cells are only like partially dyed. So if you're wondering like if it's a harsh cutoff or if it, blends into the cells, you can just take a look and see exactly what it looks like. There's a gradient, and literally only whatever's caught up in that gradient is dyed. It's kind of interesting. I'm just reducing the contrast on the screen to see if it makes it any easier to visualize, but I'm not sure if it does. It gets quite overexposed on this screen. So I'm just noticing other areas where the lymphocytes are present, and soon I'll be taking it over to the other microscope because we'll be able to see much clearer. So here we go, just putting it on the kind of microscope bed there and just kind of carefully applying a cover glass to it, and then afterwards I'll push it into the housing where it will be locked into place. Now from here, I can move the bed to try and focus it. It's got quite a long way to travel to start with, and then we move to the fine adjustment wheel, which is the more outer part of the wheel where you can move it much more subtly, because obviously you can imagine when looking at the microscopic scale, the focal plane is going to move a lot in response to very tiny movements. So here we go, I'm locking on, and we can see these cells appear, and you can still see their biconcave nature. So you'll see that they're circles, but they have like these light dips in them. And that goes to show that the cells have maintained their shape through the isopropyl fixation, and they have not burst when being stained with the methylene blue and distilled water. What I'm doing here is I see a lymphocyte, so I'm doing my digital hyperzoom, which is on top of the 40 times objective magnification. So that dark blue circle is a lymphocyte cell of a kind. We see the lovely shape here for the erythrocytes, and I'm playing with the aperture a bit as well. So things actually become a bit more blurry when there's too much light. And I suppose one way of thinking about it in terms of blender, is that if you're applying subsurface to a material, you may notice that the light bleed makes the object look a bit blurry because there's so much of that bleed happening. And something similar is applying here. The more light you provide from every direction, the blurrier, in quotes, the object will look, but you start to get used to like playing with these different dynamics. So in the thicker areas, I'm noticing that I'm really only seeing lymphocytes. And what I'm looking out for are other types of cells, like neutrophils or monocytes, which, some of these lymphocytes may actually be monocytes. Sometimes I have trouble distinguishing between them if it's not clear. And one thing to keep in mind here is that because the sample is still drying, it's not as clear as it could be. Now, I think I've noticed one neutrophil, and which tends to have like a multi-lobed interior to it. But otherwise, a load of potential lymphocytes, which they don't have much excess cytoplasm around them. So the membrane will be very tightly packed with the nucleus. But it is interesting having a look around. Like when I did this for the first time, when it was successful, I was, very excited because, again, I hadn't been trained to do this, and now all of a sudden I have the capacity to do this within my own home. So maybe I could be a little bit proud of this bit, but I came up with this technique. I'm sure other people have done this already, like come up with it on their own, but I was specifically excited because I thought, hang on, what if we oversaturate the blue values and then up the contrast on that and do like a chroma key for the blue values and then reduce the saturation afterwards? And what I found was by doing that, we can effectively heat map the color of the methylene blue staining the leukocyte nuclei. And what that does is it can make it easier to find things like the neutrophils in dense masses of erythrocytes like this. So having it zoomed out and then doing this kind of like, it's almost like thermal vision for leukocyte nuclei that have been stained. It makes it a lot easier. So here I'm pretty sure that I've found a neutrophil that I'm looking for. So it gets me thinking that this is a child sample. This is the smallest number of neutrophils I've ever seen looking in a blood smear. Now, I didn't know anything about do children have fewer neutrophils than adults? And I look it up afterwards and the answer is apparently yes. So that's interesting, although there are obviously conditions that can be involved in that, and it can vary based on whether they've had an infection and, all sorts of other factors. But this is kind of how I work really with learning things. I don't want everything to be professionally taught. I want to figure things out for myself, much in the same way that, my attitude towards creative work works as well. I see another neutrophil there down below. So this is a case of like making an observation, like doing real science myself and trying to learn something on my own. Like I noticed there are fewer neutrophils in this child sample. Is there a reason for that? If yes, then I've learned something. Okay, you might find this interesting. I accidentally let the pipette touch the microscope slide earlier, and I think I found the area of damage. And you can see here that it's literally sliced out of the cells. So again, it always strikes me as weird, like before you look at things microscopically, you think, oh, maybe there are millions or billions of things I just destroyed by letting that touch the slide. But you can actually literally just see it. You can see the exact damage that happened. And it's really not as bad as you think. Like red blood cells are actually, they're kind of big. You find that a lot of things in biology are not as small as you think they are. Anyway, that's the little introduction into the lab corner.